Good morning, everyone. I am going to start with this topic, polymorphism in man, and the chapter is genetic polymorphism. And before proceeding on to it, let's review the basics we have learned so far. So, in order to know about any organism, the best way is to disintegrate that organism into equal and distinct parts. So, this disintegration is going to lead us to all the equal and entitling entities. So let's say if you first disintegrate an organism into equal and distinct parts, you will get organs. फिर organs को disintegrate करने के बाद you will get tissues. The disintegration of which will lead you to cells, and in cell there is a nucleus. In nucleus you will see chromosomes. Those chromosomes are the condensed form of chromatin, and when you unwind it, you will get a double helix. And when you straighten it up, you will get the structure of DNA. Now, uh, let's have a diagrammatic representation. So this is cell, almost spherical in shape, and in there is a nucleus. Now chromosomes, they look something like this. When you further unwind it, these layers, they will be peeled off. So those peeled off layers will look like this structure. When you further peel them, you will get a double helix structure in this shape. And when you straighten this up, this double helix is nothing but a skewed ladder. If you hold a ladder by head and tail, and in that way, just suppose this is a ladder, you hold it by the upper hand and the lower hand. Now you rotate the upper hand in clockwise direction and the lower hand in anti-clockwise direction, and in that way you will get a twisted ladder in this form, which is known as a double helix. So this is the structure of DNA, and in the same way, when you straighten the double helix structure, it will appear like a normal ladder. So in this normal ladder, these parallel strand, the parallel handles, they are known as sugar phosphate backbone. Sugar phosphate backbone, and these middle rungs, they are nitrogenous bases. And these nitrogenous bases, which in DNA are A, T, C, G, they are nothing but the repetitive units. Because if you cut out a section of a DNA just for template, जैसे आपने ये section निकाल लिया, if you cut it out, this will appear not like this. C, G, nothing but repetition of unit in this way. So अब question ये है कि how this repetition of section will gradually lead to formation of an organism, क्योंकि we are disintegrating. Last जो equal distinct part है, we have come across the DNA, double helical structure. So if you integrate them, then you will get the organism. So double helix integrate करते ही chromatin से chromosome, chromosomes in nucleus, nucleus is cells, many cells they integrate to form tissues, many tissues integrate to form organs, and many organs integrate to form an organism. So it's a two-way road, but it's not, it's a hypothetical situation. It's not possible in real world that you go on to disintegrating an organism and you believe that after the disintegration you will be able to integrate that is the same organism into a full-fledged and live occupant. That's not possible, hypothetical, but yes, this is how this two-way road goes on. फिर now अब हम यहाँ तक आ गए that every organism is nothing but repetition of such DNA units. Now ये चेन बहुत बड़ी होती है. फिर इसी चेन में some sections they code for some genes. So let's say this section you may number this section from 1 to 8 and this from 9 to 13. So this particular section it codes for hair and this section codes for eye. So now we are looking at two distinct entities. First, this section of DNA that code for a trait. I repeat my word, section of DNA that code for a trait is known as a gene. So this section, this is a gene for hair and this one is for eye. So such sections or such genes, they combine up to form the genotypes, which is not visible. But their counterpart, the physical appearance, it is visible. So this physical visibility, it's in genetic terminology known as expression of traits, expression of genes. So gene, this section from 1 to 8, it has expressed itself in form of hair. And this from 9 to 13, it has expressed itself in form of eye. So these are the genes, the form genotype, and the visible expression, they are known as phenotype. ये अभी तक का बेसिक. फिर उसके बाद अब we have seen that let's take another example. This is repetition of unit. अब मैं यहाँ पे nitrogenous bases, I'm not writing them down. आपको समझ में आ गया है कि yes, this repetition goes on for infinity. Okay, ये repetition चल रहा है and let's number them 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17. So जो individual numbers हैं they do not mean anything in themselves. जैसे ये जो particular DNA का जो number है, is position है, 9th position, it does not mean so anything in itself. लेकिन again, let's say 1 to 7 it codes for G1, one gene. Then from 8 to 12 it codes for gene 2, and from 13 to 17 it codes for gene 3. So now these positions, these sections, they have coded for some genes, and those genes will appear in form of a visible trait, which is a phenotypical trait. Okay, अब this particular gene, इसका locus जो है 1 to 7 is the locus of this particular gene. Means hair का या जो particular gene 1 है, इसका locus इसके अलावा कहीं और नहीं होगा पूरी सीक्वेंस में, पूरी डीएनए की सीक्वेंस में, चाहे जितनी भी लंबी हो. इन द सेम वे इसका भी locus 8 to 12 के बीच में ही होगा. Locus means center, the center of operation. Now ये हो गया. फिर second we talk about meiosis. By meiosis, the gametes, which means sex cells, sperm and ova, they inherit genetic material from parents. क्योंकि meiosis, it is spatially prevalent in sexual reproduction. और sexual reproduction में, to increase the survivability of an organism, it's better that the offspring, it integrates the genetic material from two rather than one. क्योंकि दो पेरेंट्स से या दो पर्सन से जेनेटिक मटेरियल एसोसिएट करना, इट विल हैव अ बेनिफिशियल इफेक्ट, क्योंकि दोनों की इंडिविजुअल फिटनेस इट कैन वेरी, क्योंकि natural selection, it acts on individual in individual ways, in different ways. So हो सकता है कि मदर के जीन जो हैं वो पर्टिकुलर किसी ट्रेट को लेकर they are beneficial, and she holds a good stock of a good variety of survivable genes, beneficial genes, advantageous genes. In the same way, father भी किसी न किसी aspect में he may hold some better genes in terms of strength, in terms of muscle dexterity, in terms of binocular vision, in terms of hair texture, in terms of enamel rigidity. तो एडवांटेजियस ट्रेट्स दोनों में होते हैं और रिसेसिव भी होते हैं, डिसएडवांटेजेस ट्रेट्स भी दोनों में होते हैं. सो हियर, यहाँ पे जो मेंडेलियन लॉ है, स्पेशली ड्यूरिंग मियोसिस, द traits, they are assorted, randomly assorted, which means meiosis, it takes one copy from each of the parents, और वो कॉपी जो randomly assault होगी और randomly inherit होगी, that will be the best version possible for the survival of organism. So this is the way nature works. This is the way whole DNA replication works. So this is a system of DNA replication by the process of meiosis. So यहाँ पे ये assume कीजिए कि यही DNA replication, it tries to integrate or try to inherit by replication the best traits possible. So दो से traits आते हैं, they are always beneficial. Heterogeneity is always beneficial, is always gets an upper hand than homogeneity. क्योंकि ये बिल्कुल आप समझ सकते हैं, हर कोई, it's a no brainer thing that if individuals are different, they have more chances of survival. And if individual are homogeneous, then any drastic event or any inclement event will either save them completely or extinct them completely. ये बिल्कुल आप समझ सकते हैं. उसके लिए let's take another example. For example, this is a piece of land, और यहाँ पे ये डिफरेंट तरह की वेजिटेशंस हैं. सो वी कैन से the vegetation of this particular piece of land, it is heterogeneous. अब यहाँ पे अगर any inclement event like wildfire, अब wildfire जो होगी, it rages on, और हो सकता है the sum of them, जितने भी vegetation है, some of them they burn down and they become extinct. लेकिन कुछ varieties जो हैं, for example take this one, अब हो सकता है कि it gets over, it is not harmed, and this one also it is not harmed. इस situation में ये जो तीनों चारों species हैं who which are saved from the wildfire, they will live up and proliferate by the way of reproduction, by which their offspring will survive. क्योंकि वो अगर wildfire survive कर सकते हैं, then any another such instance of wildfire will be potentially disarmed by their own genetic material. And ये जो species हैं which go extinct, which which could not stand up to the wildfire, they become extinct. अब let's suppose यही situation homogeneity की होती है और सारा का सारा सारी vegetation है it's of one type, which means homogeneous. अब कोई भी inclement event, let's the same example wildfire. A wildfire में either ये पूरी की पूरी species है, it can hold itself against wildfire, या तो पूरी की पूरी species survive करेगी या पूरी की पूरी species it went into extinction, it goes into extinction. So that's the advantage of heterogeneous heterogeneity. So in that way, genetic material, it will be copied by both the parents. So in that way, every offspring will have two copies of any particular gene. So ये existence of two copies, it is known as alleles. जीन सेम है, जीन वही है, मे बी G1 फॉर आई बोल सकते हैं, बट इट विल हैव टू वेरिएशंस, टू फॉर्म, बाइनरी फॉर्म, क्योंकि एक कॉपी पेरेंट से, एक कॉपी मदर से, और दोनों कॉपी सेम भी हो सकती है, दोनों कॉपी अलग अलग हो सकती है. मतलब होमोजीनस भी, इट कैन बी होमोजीनस, इट कैन बी हेट्रोजीनस. ये हो गया. अब लेट्स कम टू पॉलीमॉर्फिज्म. पॉलीमॉर्फिज्म is in its literal sense means existence of multiple morphs, and a morph is variety of alleles. So this variety of allele, this is known as polymorphism. तो उसके लिए कुछ कंडीशन है. फर्स्ट, सेम लोकस होना चाहिए. सेम लोकस पे नहीं होगा, देन इट मींस दिस इज अ डिफरेंट जीन. फिर दोनों जीन को, however similar they look, we cannot count them as a condition of genetic polymorphism, or a condition of being genetically similar. फिर सेकंड कंडीशन है that it must be available at least in 1% of the population. So अगर वो वेराइटी उस ऑर्गेनिज्म की जितनी भी पॉपुलेशन है, अगर ह्यूमन की बात करते हैं, वी आर 8 बिलियन, देन इन दैट केस 800,000 or 8 million. Yes. So in human population, if you want to say that for a particular locus G1, there are three groups of population: Caucasoid, Mongoloid and Negroid. ये बड़े बड़े एग्जाम्पल इसलिए रहा हूँ ताकि it will be better to understand. So let's say G1 में, सेम G1 की जो पॉपुलेशन है उसके अंदर the binary forms are like this, alleles are. Then same gene G1, it manifests in यहाँ पे in terms of Caucasoid, it may be available in this form. फिर Negroid की या Mongoloid की बात करते हैं, it may be in form of G dash D dash, and in form of Negroid it may be G star. So now we have seen कि in different population the locus is same, gene is same, the section it code for वो भी same है, but the variety they are different. वो variety हो सकती है natural selection की वजह से. So the same gene, it has different varieties in different population, and all these variety they count up more than 1% of population, means more than 8 million people. So when existence of such a trait is identified, this condition will be known as genetic polymorphism. Now let's come back to the chapter. So polymorphism in man. So first instance is about sickle cell anemia. which is genetically controlled by a gene which produce the disease when homozygous and responsible male for sickling trait when heterozygous. This gene affects the formation of hemoglobin, but not for all. It only affects the structure of erythrocytes that resemble a sickle-like shape that leads to hemolysis, which is severe enough to cause an extreme and often fetal anemia. अब इस पैराग्राफ का मतलब ये है that normally erythrocyte means red blood cells. Red blood cells, they are spherical in shape, but due to sickle cell anemia, in some population, these cells, they take up a form of a sickle. This one, not they do not exist now in spherical form, but in a form that resemble a sickle. अब इसकी वजह से the person may suffer from hemolysis, which means anemia, loss of blood. And as you know, loss of blood is always lethal, especially when it does not coagulate especially when it does not lead to clotting. So ऐसी situation में this the presence of the sickle cell anemia, it may prove lethal. But साथ में ये चीज और कही है, which is that this gene RBC, जिसके लिए जो gene है particular hemoglobin, so when this is in spherical shape, this is known as HGS, which is its locus. Ab when it takes a sickle shape, it takes the variety of HGA. अब ये दोनों इसके alleles हो गए. अब 3 conditions होती है. First is homozygous dominant, then heterozygous dominant, and homozygous recessive. अब लेट्स से एक पॉपुलेशन है जिसके अंदर दोनों डोमिनेंट है, HGS, HGS. अब यहाँ पे तो स्पेरिकल शेप ही है, तो homozygous dominant, it won't be affected. Then second condition, HGS, HGA, यानी dominant for spherical shape and recessive for sickle shape. अब heterozygous है, but as you know, nature favors the dominant. So in that case, the spherical shape है, it will be manifested, और जो population, जो individual है, it may have some disease traits but in phenotypically, the presence of recessive gene along with the dominant one, it will not have any impact on the health of the heterozygous individual. So that individual is going to be perfectly healthy. अब homozygous condition देख लेते हैं जिसमें दोनों ही sickling traits हैं. In that condition, this recessive homozygous recessive, it will be lethal because of hemolysis. So ये यहाँ पे बात की है. So it must be noted that the anemia is recessive while the formation of abnormal hemoglobin is not. So यहाँ पे traits की बात कर रहा है, which traits are recessive and which are not. So anemia, it is recessive because recessive genes are those which do not help in the survival of individual and needs elimination or deletions. So anemia, it's as you can see, anemia is never going to be helpful in the survival of a species. So this will be eliminated. That's why it's recessive. So, and then the formation of abnormal hemoglobin is not. So if spherical shape, it is taken up by a sickle shape, so this change in shape will not lead to anything. But if this change in shape, if it leads to anemia, a loss of blood, then that's going to be problematic. The same genotype or phenotype का जो है, that genotype are different and phenotype are different. जैसे अपन heterozygous condition में हमने देखा है कि यहाँ पे spherical and sickle shape दोनों हैं. So this individual will be heterozygous for this hemoglobin, but Since heterozygous are dominant gene, so the expression of dominant gene will be ascertained. So in that case, उसके blood में there will be sickling traits, but the principle of natural selection, it says the phenotype, it will inherit the best one possible, which is the spherical shape. In that case, even if the individual holds a sickling trait or a recessive trait in his genotype, that gene will not be expressed. Then the heterozygotes, they are quite common in certain regions of European population, in Greece, Italy, in African tribe, and even in India. So evidently, अगर इतनी population है, then it must have an advantage which strongly counterbalance the destruction of homozygotes, and which it means कि अगर इतनी सारी population में this heterozygous gene is present, this line is talking about this allele. कि this allele is present in that large population. So अगर अभी भी present है, then there must be reason, and वो reason है that it has certain survival advantage over the homozygotes, which are the individual which holds both the genes as recessive. So the area that I've been talked about are the area that the sickle cell trait that confer marked immunity against malaria due to Plasmodium falciparum. So the polymorphism is established only in places where malaria is common. So again, so वो reason हमारे पास आ गया कि आखिर नेचर ने heterozygous जो है वो कंडीशन क्यों establish की? Because this change, this sickle trait, it helps in the population. It helps those who are suffering from malaria, who get diseased by this particular vector, Plasmodium falciparum. So if it affects your immunity, you are going to die. So sickle trait, it provides an immunity, but it provides an immunity only in heterozygous form. If it stays in homozygous condition, it leads to your death in form of hemolysis or anemia. So that's the gist of it. I repeat again that the presence of sickling trait is necessary, especially where malaria is endemic, because this vector, Plasmodium falciparum, the vector agent of malaria, this causes the blood thinning. And in that case, an individual, it must shrink the shape of the red blood cell so as to preserve the blood. But since the accumulation of or the presence of both the alleles in form of sickling trait or in the form of sickle, they will lead to anemia somehow. Even if you do not die from malaria, you will die from anemia. So here the advantages are conferred by the presence of one dominant gene, because nature selects the dominant. In that case, the spherical shape will be there, the appearance or the phenotypical expression will be of spherical shape. But since your gene, your genetic material, it holds a sickling trait, this sickling trait will provide you immunity against the vector of Plasmodium falciparum. So this is the ultimate gist. Then let's talk about another condition, thalassemia. So the condition was discovered by Allison in 1954, that many homozygotes, perhaps a few heterozygotes, die, yet the gene is present in 10% of population in Greece and Italy, where malaria is endemic, and India as well, particularly in parts of central India where many tribes inhabit. Such polymorphism exists where sickle cell anemia is found to be prevalent in malaria endemic regions. So thalassemia and sickle cell anemia, both of which they are prevalent in malaria endemic regions. So again, the concept is same, that to prevent death via malaria or thalassemia, there need to be a change in the genetic material, that at least one heterozygous should be there, one dominant trait should be there. If both are recessive, this one is dominant condition. So there are two instances of dominant condition. First, when the binary form, both the alleles are of dominant nature, and if there is one allele of dominant nature, both will provide an immunity, both will be expressed. But if both the genes or both the alleles they are of recessive nature, then it will be lethal in itself. So instead of providing immunity against the the disease for which the recessive took place, for which the genetic form arrived, it will kill the individual because the presence of both the traits. Anyway, एक ही एक चीज बार बार आ रही है, आप ओवरऑल तो इसका समझ ही गए. Then one of the oldest known such polymorphism is the ability to taste phenyl thiocarbamide, which is PTC or phenyl thiourea, PTO. So for some people, PTC only has a faint taste or no taste at all, but for others, it has a very bitter taste. So this tasting, it is controlled by a single dominant gene T that determines a high sensitivity for the taste of PTC. Then the non-tasters, they are homozygous for the recessive allele tree. When both the parents are non-tasters, their children will be non-tasters. But when parents is taster, other is not, this heterozygous condition again okay. So here PTC again is a genetic polymorphism. So PTC, this particular gene or this particular trait is taken into account because it is the taste of PC, ability to taste the PTC determines how much a population can taste different ingredients. So now this ability to taste, it is determined by gene T. अब आपको पता है कि कोई भी ऑफस्प्रिंग जो है, it can have three thus forms. First, both are dominant, then heterozygous dominant, and then recessive homozygous. ये तीन कंडीशंस हैं, तीन फॉर्म ऑफ अलील हैं. अब ये दोनों जो हैं ये heteros हैं, and because of their very nature, the dominant will be expressed, the recessive will be masked out or covered. In that case, this will not, the presence of one recessive will not have any marked effect on an individual. But if both are recessives, especially in this case, both are homozygous, this will have some clear expression, either in form of disease or particularly in case of PDC, it will manifest itself in form of no taste at all. So इन दोनों की बात करें, they will taste any substance, even if the gene for no taste is there. Small t means no taste and capital T means taste. So they are able to taste a wide variety of substances, including the bitter one. And this trait is favored by this heterozygosity. It is favored by nature. So these individuals, they have conferred immunity than that of the homozygous condition. फिर ये लाइन when both parents are non-testers. So ये condition will not be difficult to understand. As you know, an offspring inherits one copy each from mother and parents. So अगर parents खुद ही, if उनका genetic makeup उनके parents से, they have inherited recessive for PTC, which means small t. दोनों जो allele हैं, they are small t. In case यहाँ पे meiosis process किसी भी random assortment से वो जो, क्योंकि random assortment जो होता है this way, 4 varieties. ये मदर की साइड है, ये फादर की साइड है. अब this multiply by this derive this one, this multiply by this derive this one. ये मेकअप, ये और ये. तो अल्टीमेटली जो जो इनहेरिटेंस पैटर्न है ये सिमिलर होगा, है ना. सेम रिसेस ही भी डिराइव होंगे. आप दूसरी कंडीशन ले सकते हैं, अगर एक मदर जो है शी इज रिसेसिव एंड फादर जो है वो हेटेरोजाइगस डोमिनेंट है. इन केस अब इन दोनों को अगर मल्टीप्लाई करते हैं टी तो एक ऑफस्प्रिंग जो है वो हेटेरोजाइगस डोमिनेंट होगा. फिर सेकंड ऑफस्प्रिंग जो है वो होमोजाइगस रिसेसिव होगा. देन थर्ड चाइल्ड will be heterozygous dominant and fourth child will be homozygous recessive. So in that case उनके 4 ऑफस्प्रिंग में से 2 तो होंगे रिसेसिव मींस नो टेस्टर्स एंड 2 होंगे डोमिनेंट, यस डोमिनेंट वन, और वो होंगे टेस्टर्स. ये है नेचर का सॉल्यूशन to deal with the issue of survival. Then genetic polymorphism may be transient or balanced. Genetic polymorphism are balanced if selection favor the heterozygous. जो conditions हमने देखी थी पीटीसी के केस में हो गया, सिकल सेल अनीमिया के केस में हो गया, थैलेसीमिया हो गया. In all such cases or many such instances are there in which heterozygotes are favored or natural selection favors heterozygosity, because in that way heterozygosity is condition of presence of at least one dominant trait, and according to the nature of genetic inheritance that dominant trait will be expressed. And since that trait is called dominant because of its survivability, the expression of which ultimately lead to survival of the offspring or the individual and ultimately the whole population. So that's why the balanced genetic polymorphism is the favoritism of heterozygosity. When selection favors the heterozygotes, a stable equilibrium may be achieved and a substantial frequency of both allele may be maintained in one environment. So again, this is a good hypothesis कि अगर heterozygous रहेंगे तो ये जो condition है this can lead to a stable equilibrium, stable condition of equilibrium, क्योंकि यहाँ पे डोमिनेंट एंड रिसेसिव दोनों जीन एंड दोनों की फ्रीक्वेंसी अल्टीमेटली it will help in the maintaining of stable count of population. So this balanced or stable polymorphism is a result of natural selection operating as a stabilizing agent. So यहाँ पे जैसे कहा है कि natural selection ही है जो select करता है. Otherwise we by ourselves we do not artificially select uh, the dominant gene for PTC or thalassemia or कोई भी condition हो, we do not artificially select it. It is ultimately selected by nature by process of meiosis. So this is it. Thank you very much. फिर next chapter में we will go to Hardy-Weinberg law. Thank you very much.